It's okay.
Nabila.
Can you hear me?
Yes, I can hear you. Okay. So Nabila, the floor is yours.
Thank you very much, Fajar, and everyone for the nice introduction. And I would like to start first by thanking Dr. Ratim and Dr. Fajaruddin for receiving us here at BRIN and for giving us the opportunity to share some of our work on banana. Thank you very much. So I will start by a brief introduction about where we come from. So we are both, and together also with Guillaume, a research scientist at CIRAD, and CIRAD is a French institution for agricultural research and international cooperation, working for sustainable development of a tropical and Mediterranean region. And we are based in France, in the city called Montpellier, which is in the south of France. Guillaume and I work in a research unit, which is a joint unit which comprises researchers from different institutions. And these are researchers from CIRAD, but also from the National Research Institute for Agricultural and Environmental Research. And there are also two educational institutions, Institut Agro of Montpellier and the University of Montpellier. So the research unit is called AGAP Institute for Genetic Improvement and Adaptation of Mediterranean and Tropical Plants. So Guillaume and we work in a team called Structure and Evolution of Genomes, a SEG team, and our head of team is Dr. Angélique Dont. And we work on two model studies with complex genomes. So these are banana on the left side and sugarcane. And we not only work on these plants because they have they're important on the social and economical point of view, but also because they have specific feature that they have complex genomes. Which means that they are polyploid, so they have more of two chromosome sets in their genome, which is the usual case for diploid organisms. And they also result from interspecific hybridization, so hybridization between different species or subspecies. So that's the case for banana here on the left side, which has cultivars with different ploidy levels, 2, 3, or 4 times the normal chromosome set. And also on sugarcane, the genome is even more complex with something like around 12 copies of each chromosome. So our main research question is the understanding of the architecture, the diversity, and the evolution and the genetics of these genomes. And we're interested in that to provide knowledge, to provide information and tools to support. Is there a problem? Everything okay? Yeah. Is everything okay? Can I continue?
Yes, everything is okay, Nabila.
Okay.
Please continue.
Sorry, I just heard a voice. I wasn't sure. Okay, so we do that to provide information and tools to support and facilitate breeding. So the main research questions are around genome architecture and diversity, and this will help us to knowledge about this will help us to understand better the origin, the evolution and domestication process of these crops, and also to elaborate strategies for the use of genetic resources. We are also interested in characterizing the genome architecture and its impact on chromosome segregation. And this also will help us to understand the evolution of these species and to elaborate breeding strategies. And we also have researchers working on the identification of genes and genomic regions that are involved in diverse agricultural traits, including disease resistance, for example, or food quality. And this, of course, will have an impact on the design of breeding strategies. So today, and we also use different strategies and methods, starting from gene and transposable element annotation, genetic mapping, marker linkage, genome-wide association, and structural comparative genomics using reference genomes and resec data, And we also have a cytogenetics lab in our team. So we produce genomics resources, and we also develop bioinformatic approaches to analyze this genomic data. So I will start then by one of the main research questions we have been working on these last years, which is the characterization of the banana mosaic genomes. And we do this to try to learn from the genomes about the origin of cultivated bananas. And I will also present some information about how this information is important for breeding new bananas. So few element of context that probably many of you know about is that banana, I think most of you know that it's a staple food essential for food security in many countries, and also an important source of income from smallholders, with two main uses, dessert and cooking, and a locally existing diversity in Southeast Asia. While for export dessert bananas, it's rather a monoculture of clones originating from one cultivar, which is the Cavendish cultivar. And this cultivar accounts for more than half of the global world production. Sorry for the slides. Okay, so what do we know? Previous scientists have worked on the understanding of the origin and diversity of cultivated banana. So there are two main species who are involved in the formation of banana cultivars, Musa balbisiana and Musa acuminata. And Musa acuminata has diverged along Southeast Asia in different subspecies. Some of them are depicted here in this map. And the hypothesis is that the domestication involved hybridization between this species and subspecies, which was made possible by human migration. So there was contact between these species and subspecies that were previously isolated. And there was also selection of diploid and triploid, seedless and parthenocarpic hybrids by early farmers. Okay, so the supposed domestication process starts from different subspecies from Musa acuminata with different AA genomes. So A is the Musa acuminata genome, and also Musa balbisiana. And following intersubspecific hybridization and selection for parthenocarpy and seedlessness, diploid cultivars were formed. And in other cycles of hybridization, there was polyploidization that was due to the production of 2N gametes in some cases. And this resulted in the formation of triploid cultivars. After that, some of these triploid cultivars and diploid cultivars, they were selected by humans, and they were vegetatively propagated for centuries because, of course, they were rather sterile or poorly fertile. And these centuries of vegetative propagation resulted in somaclonal diversification. So that's another level of diversification of banana cultivars. And it is also supposed that the irregular meiosis suggested that there was cytogenetic studies carried on, and irregular meiosis observed during these studies suggested that there were structural variations which may have favored reduced fertility and transmission of 2N gametes. This monoculture of somaclonal variant is highly vulnerable to disease, and among them, fusarium wilt, of course, and most of you know maybe that a new strain, tropical race 4, is spreading globally and destroying Cavendish plantations, and it's also a danger for many local varieties, and there's no efficient treatment for this disease. There's also black cicatricea, and Jean Carrier will talk a bit more about this disease. Black cicatricea requires quite heavy pesticide treatment. So there's an urgent need of improved resistant banana cultivars, and these can be produced based on mainly two different breeding schemes. First one where people exploit the residual fertility in triploid cultivars, and so the triploid is crossed with an improved as a cultivar or a wild improved diploid with targeted traits. And this will result in a complex progeny with different ploidy levels. People will select tetraploids that are then crossed into diploids to generate a triploid cultivar at the end. The other scheme, reconstructive breeding, it goes through a tetraploidization of diploid cultivar, usually by colchicine doubling, and then one crossing step with a diploid individual to generate a triploid cultivar. And to be able to be more efficient in performing these breeding schemes, it is necessary to understand better the origin and genomic architecture of major cultivars. So that's why we were started to investigating the mosaic genomes of these center-specific hybrids. And so what do we mean exactly by the mosaic genomes? If you have a first hybridization between two wild individuals, as represented here in this schematic scheme, the first generation of hybrid will just have a combination of both genomes. And if there are further hybridization cycles, or for example, a backcross to one of the parents, then you start to have recombined chromosome in the next generation. And then when another hybridization follows with another species or subspecies, then the genome starts to be more complicated with additional contribution, and so on with different hybridization with different species and subspecies. And at the end, you end up with individuals where the chromosomes or different segments along chromosomes have different wild ancestral origin. And the question we ask is, what are the ancestral origins along chromosomes in cultivated banana genomes? So we do that. We were able to perform these studies because we had banana reference genome sequence assemblies available. First one from Musa Caminata malacensis was published in 2012, and it was recently improved with the new sequencing technologies and published in 2021. And this version is much more complete, and that's the one we are using currently for our studies. And this was done with our collaboration with the French sequencing center Genoscope. So to do the study, we also sequenced at the whole genome sequencing level a set of around 150 accessions from wild and cultivated banana. And this was done with Illumina short read sequencing, also in collaboration with Genoscope. And in this setting, we had few representatives from some of the main species and subspecies that people already found that they contributed to the genome of cultivated banana, or that were representative generally of the diversity of banana. We also had diploid cultivars and also triploid cultivars from different parts of the world. We had to develop methods to infer these genome ancestry mosaics, and I will not go into the detail of the methodologies. These methods had to be developed because there were several constraints. So, for example, the presence of introgression in wild representatives of Musa Caminata, distinct level of fixations, and possibly not all ancestral groups were represented. We also had a mixture of diploid and triploid. So Guillaume Martin and colleagues developed methodologies based on multivariate analysis and private allele approaches. And the idea was to identify alleles that were specific to each of the wild ancestor that we wanted to test as a contributor to the genome of banana cultivars. And we use these specific alleles and information on their coverage to perform chromosome painting, and the final result looks like this. So that's an artificial representation of banana chromosomes. And here we have the 11 banana chromosomes. So that's a hybrid banana cultivar represented here. And for each chromosome, we have two bars that represent the two haplotypes. And different colors represent different wild origin. And so, for example, we know that in this region of chromosome 1, we have the blue origin and the green origin. And when we look at the data set that we analyzed, the results showed us that we had quite a diversity of genome mosaics among cultivars. So we had here diploid cultivars with 3 ancestral contributors, and you have here the legend of the relation between the color and the species and subspecies that we tested and which we found as contributor to cultivars. And the simplest mosaics, they had 3 ancestral contributors. That's the green, red, and light blue color representing Musa banksii here, Musa acuminata zebrina, and Musa schizocarpa. Sorry, it's a bit of a problem with the computer here. Other individuals had more complex mosaics with more contributors. For example, in the middle here, 5 ancestral contributors, one of them unknown. And here is an individual with 6 different ancestral contributors, 2 of them unknown. So from these observations, we can see that the mosaics were much more complex than anticipated. So we had several generations and meiosis at the origin of these cultivars. And we identified 8 main ancestral groups that contributed to the majority of the cultivars that we analyzed. And these ancestral groups include the species Musa schizocarpa from New Guinea and 2 unknown groups for which we didn't have a pure wild representative. So if we look at the distribution of these cultivars, the diploid ones, the simplest mosaics that we observed were from accessions that were prospected in Papua New Guinea with 3 ancestries. And then we also found that these 3 ancestries that we found in these simplest accessions, banksii, zebrina, and schizocarpa, they were present in all analyzed diploids. But then if we look at the different other cultivars, we see that the mosaics show different color patterns, which means that there is quite a high diversity in these genomes mosaics, and that other hybridizations occurred with local wild Musa species and subspecies along the different geographical area from which the cultivars are originated. If we look at triploid cultivars, they also show contrasting mosaics as depicted here for some of the major. So here we have the mosaic of a Cavendish cultivar, and this here is plantain, for example. And so we have really contrasting mosaics, which means different ancestral origin. However, when we look at the mosaics, we always find these 3 colors, the green one, the red one, and the light blue one that represent the banksii, zebrina, and Musa schizocarpa ancestry. So what we did, because although we expected the presence of some banksii and zebrina contribution, we didn't expect that there was such a widespread contribution of the species Musa schizocarpa. So that was quite new. And so we looked at the introgression breakpoint, which means the place in the chromosome where the origin shifts from Musa schizocarpa to another origin. So we selected several of these breakpoints, which were present on the diploid cultivars from Papua New Guinea, so the ones with really the simplest genome mosaics. And we actually could show that at least one of these Musa schizocarpa introgression breakpoints was conserved in other diploid and triploid cultivars from all over the world. So we concluded from this that this pattern of introgression of Musa schizocarpa, they indicate a shared origin, a common origin of banana cultivars. And this, because Musa schizocarpa is only found in the New Guinea region, this suggests that the origin of this shared origin is from the New Guinea region. And after that, these cultivars were likely transported along Southeast Asia, and they were introgressed with local wild Musa. So this shows the hypothesis that we have about the origin and the diversification of banana cultivars. And there was likely many movements of cultivated and wild materials in this region. The other thing that we could also find from this work is the contribution of unknown ancestor to important cultivar. So this is the mosaic of the Cavendish banana. And the pink color that you see here, it corresponds to a wild pool, which we could not identify as of yet. So we didn't have a wide representative for this ancestor. And there is also probably a smaller contribution from another unknown genetic pool. And so these unknown ancestors, they are present in important cultivars. And we believe that it's really important for breeding and for a better understanding of banana origin and domestication to characterize these unknown ancestors. So we did some phylogenetic analysis to know a bit more about where these two ancestors could be positioned phylogenetically. And we could show that they are within the Musa acuminata Musa schizocarpa group. And the painting of accessions in public databases suggested that the unknown ancestor one could be Musa acuminata subspecies halabanensis. The second contributor we still don't know of, but the distribution of cultivar with this contribution points to the island Southeast Asia as a first starting point to search for this unknown contributor. So what we also did is, so we tried to understand the origin of banana from the wild ancestor, and we also studied at another level what could be the pedigree relationship between current diploid and polyploid cultivars. And so Guillaume Martin, he designed a methodology to analyze these pedigree relationship, and he could show that the Mshahe diploid cultivars that are nowadays only found in East Africa, they contributed their complete genome to several important dessert banana, such as Gros Michel and Cavendish, but also other groups such as Lendra Padassi, so AAB banana, and Pome and Nadan. So this diploid was very important in the origin of important cultivated banana, especially for dessert banana. But it also contributed its genome to 1X gamete to diverse diploid and triploid cultivars that are shown here. So Mshahe was involved in the origin of important dessert bananas, but was also involved in several other crosses. So after that, we also characterized the large structural variation in wild and cultivated banana. So the presence of this structural variation was already predicted by classical cytogenetic studies, like the work of Dr. Shefford, for example. And our objective was to characterize these large chromosome structural variations that are present in subspecies informed in the formation of cultivars, and to determine their impact on recombination and segregation of chromosomes. And this was done in the following way. It was based on the study of genetic linkage between markers within hybrid progenies and comparison to the Musa acuminata reference genome. And this graphic here, it depicts the linkage between molecular markers along chromosomes. So these are the 11 chromosomes of the, based on the 11 banana chromosomes, sorry, from 1 to 11. And the coded color is the less linkage there is, and the hotter the color is, the more linkage there is. And what we can see here is that there is linkage between chromosome 1 and chromosome 4. And this suggests a translocation involving these two chromosomes. And this also results in high segregation distortion and reduced recombination in the area that are impacted by this chromosomal translocation. This was confirmed by cytogenetics and by genome assembly. And so we could describe more precisely the reciprocal translocation between chromosome 1 and 4. So these are the reference chromosomes, chromosome 1 in the reference, chromosome 4 in the reference, and the structure in chromosome 1T4 and chromosome 41. And this structure is present in the Musa acuminata subspecies malacensis, but only in some of the accessions of subspecies malacensis. In total, when studying all the different subspecies that we had, there were 7 large reciprocal translocations identified and 1 large inversion. And looking at the distribution of this translocation, so the ancestral structure of the genome is found in the Banksy and Musa schizocarpa. And then the Zebrina bears chromosome 3A translocation. Some of the malacensis bear the chromosome 1,4 translocation. Some accession from Biomanica, they bear 2 translocation between chromosome 1 and 9 and 2 and 8. And in few of them, they also carry an additional one between chromosome 7 and 8. In the unknown genetic pool that we don't know to which wild species it corresponds, there is also a large chromosomal translocation between chromosome 1 and 7, and also between Musa acuminata and Musa balbisiana, there is a chromosomal translocation and inversion. So the distribution of this large structural variation actually corresponds to the subspecies and species classification. And we also found that these translocations, or some of them, are found at heterozygous states in many cultivars. At least 60% of analyzed cultivars contain or bear these translocations. And they're also found at heterozygous state in most parents of the breeding program. So what would be the impact of translocation, for example, on banana genetics? So for example, on QTL analysis, there was a study by Stella Babiani, it's published in 2021, on QTL for food quality in a progeny from Pisang Madu crossed with Galeo. And Pisang Madu bears the 1 and 7 chromosomal translocation that is depicted here. And the QTL study showed that there were QTL for food quality on the translocated, linked to the translocated chromosomes 1 and 7. And it was also observed that there was no recombination in this translocated chromosome 1. So it means that we cannot be more precise about the QTL position. So that's the downside of the presence of the fact that the QTLs are present on this translocated chromosome. Because it doesn't recombine, we cannot go further in the characterization of QTL positions, but this also would have a positive side. It would favor the QTL monitoring in breeding programs. It would be easier to follow than in the breeding program. So actually, most of this chromosome of Pisang Madu, it originates from one of the unknown ancestors. And this chromosome 1T7 is present in Cavendish and most dessert type bananas that were analyzed. So in conclusion, from these studies, we think that we brought new light on banana origin and domestication by showing that cultivar genomes are complex mosaics involving several wide Musa genetic groups, that there were more hybridization steps and more contributors than previously thought, in particular Musa schizocarpa and these unknown contributors, one of which is still to be discovered. And we proposed that domestication started in New Guinea, that cultivars diversified through hybridization with wide local Musa during their transport with human migration or through trade. We also showed that a few key cultivars have played a major role in the banana expansion. So because we found this common introgression in diploid cultivars from New Guinea, and that they are present in almost all other cultivars we analyzed. And also based on the fact that, for example, the Mshare diploid cultivar is a direct parent of several major cultivars On the other way, so what would be the impact of this knowledge on breeding strategies? So on the identification of ancestral origin and parents of major cultivar, we believe that this should help developing breeding strategies based on the genetic resources and on these parents, in which we should introduce disease resistance. And this should also help guide germplasm prospection and conservation strategies. And that's why we believe it's important to characterize better, for example, the unknown ancestors through the analysis of additional collections or new prospections. And on the fact that the large reciprocal translocation are present at heterozygous states in most cultivars, and that they have an important impact on recombination and segregation of chromosomes. So this should have an impact on the design and interpretation of genetic studies to identify QTL, for example, and also on the design of breeding strategies to incorporate interesting or important characters in cultivars. All the genomic resources that we produce are available for the community on the Banana Genome Hub, and there were also some tools present to analyze these data. So, and this is acknowledgement of the people who were mainly involved in this work, in particular Guillaume Martin, which is with us here, and of course several other colleagues from CIRAD and PhD students, Marion and Aurélien, and colleagues from the banana genetics and breeding team in Guadeloupe, and also our collaborators in Genoscope and the different banana collection in Guadeloupe, and also the international collection in biodiversity, our computer facilities, and we also acknowledge the diverse funding sources that are presented here. Thank you very much for your attention. And all questions are welcome.
Okay, thank you, Nabila. It's very fascinating and interesting of the genetic of banana, the complexity of banana in cultivar, or even in the where relative itself is it's actually more complicated than we are thinking for. Okay, I open. Okay, so maybe because of the presentation of Yang, probably it's more about phytopathology. So I open 3 questions for Nabila. Everything about genetic or genomic or banana, or probably if you ask something about general genetic, is it also okay, Nabila?
Sorry, yeah, sure, and maybe I will ask for maybe also Guillaume to maybe answer some of the questions, if you don't mind.
So I open 3 questions.
Yeah.
No one?
Please feel free to ask questions. All questions are welcome. Yeah.
Please feel free. Or if you are still thinking what question that you like to ask to Nabila,
we can also
in the
have
meantime, we can continue to the okay, here you go. Burita Andini. Okay, please, Burita.
Yeah. Thank you, Mas Fajar, for the nice opportunity. I would like to ask one question to the speaker, Nabila. I read somewhere that in Asia, Indonesia and Thailand are considered as two of center of diversity of banana in Asia. So could you please confirm whether this is true, and if yes, which might be higher, the genetic diversity from Indonesia or Thailand? Thank you so much.
Yes, thank you very much for your question. Yes, indeed, in Southeast Asia, you have a lot of diversity of the Musaceae family and also of the genus Musa. Yeah, so I think in different areas in Southeast Asia, you might find different diversity of Musa. For example, there are many different Musa species in Thailand or the whole Southeast Asia continental area, but also a lot of diversity in Indonesia. That's quite clear, and I think Dr. Fajar, he also characterizes some of this diversity. So depending on the type of species and subspecies, you might find some differences, but the whole area is actually the area of origin and diversity of Musa. Yeah.
I hope it's quite clear, yeah, Bu Rita. Okay.
In the
chat. Okay. Next questions.
Fajar, can I ask some questions?
Yes, please, Pak Wicaksono. Okay, so this is one of our initiator of banana research in our institute. Please, Pak Wicaksono.
Thank you. The mosaic of those 5 or more genomes would require at least 4 sequential cross of parents endemic to different areas. So I would like to ask you, could you hypothesize how long would it take to get that cultivars with all those 5 genome mosaics from the wild? Well, the reason I ask this is that, if human migration may contribute to those recombination, then the human who bring that the first or second cross would, I believe that they have immediate use for that. The immediate use, I would believe, that would be seedless. So if this seedless would brought everywhere, to many places, and then they would cross again with some wild, and until 4 to 5 times, then since it's seedless, then it would require very long time. I think that's one question, and then the other one is in line to that the schizocarpa is also CD. Why would people bring the schizocarpa to everywhere to cross them? Thank you. That's all my questions.
Thank you very much for your question. Okay, maybe I'll start with the last one. The hypothesis that we make is not that Musa schizocarpa itself was transported. What we hypothesize is that hybridization happened in somewhere in the New Guinea island, or because Musa schizocarpa is, to my knowledge, only identified there. So we think that there were crosses, hybridizations there between Musa schizocarpa and Musa acuminata subspecies banksii. That maybe would have been the first step. As we find Musa acuminata zebrina, the simplest hybrids that we identify. We don't really know if this third hybridization was really there at the beginning, probably not. So we don't exactly know the exact process, but we think there was already hybridization there, and what was transported was already hybrid. Yeah, not the wild. That's the hypothesis that we make. That's what we propose. Okay. I don't know if this is clear. And is it okay? Does it answer your question?
Yeah. But is there any cultivar, that the remnant probably, that is cultivar that is a mix of those Schistocarpa and Banksii, that is edible and people still cultivate them?
Yes. Sorry, maybe I wasn't clear in my presentation about that. So the cultivars that were prospected in it was in Papua New Guinea. So it was the eastern side of the island. These are the simplest mosaics that we find, and it's possible that they correspond to, or their parents or relatives correspond to some of these early cultivars. So for us, the best representatives of these early cultivars would be these individuals, these cultivars diploids from the New Guinea. Yeah. So that's what we had in our setting, actually. Yeah.
Okay.
So from our setting, for these would be the closest one to the early cultivars, right? Yeah. And the first question would be if it's possible to reproduce from the wild to obtain banana cultivars, right? Yeah. Yes, I think.
Yeah.
Okay.
Could you, because it's a combination of more than 5 genomes, that there would be require crosses, to at least 4, probably 3 or 4, and the 3 sequential crosses with the wild from the seedless cultivar that people brought, that would require a lot of work and probably a lot of time. That's my question. Could you hypothesize how long it would take from the wild become cultivar that have those 5 or 6 mosaic?
Yeah, okay. I can't really hypothesize on that, to be honest, because one of the main traits that breeders have to obtain at the end is a seedless parthenocarpic fruit. And you can only have this trait if you use one cultivar in your breeding scheme. Okay. To my knowledge, I don't know of anyone who has been able to reproduce this trait by crossing the wild subspecies, right? And yeah, exactly. So I think it would be important to know more about this trait in banana. I think it would be interesting to know for the origin of the parthenocarpy and seedlessness in banana. It's probably, yeah, it's possible that and that would also explain why we have such common regions shared by cultivar. So maybe something happened in these early first crosses, that was really gave interesting trait, possibly the seedlessness and parthenocarpy. So that's a hypothesis, yeah. And something happened there in these first crosses. And right now, I'm not sure it was possible to reproduce it, but the more we know about the genome structure of cultivars, the more we can think of devising schemes to maybe try to start from the wild. But it would be a lengthy process with the current knowledge that we have. With the current,
exactly, because if crosses involving a parthenocarpic, that would be difficult, and yet you have those 5 mosaic. I was thinking, how long would that take, to get all those? Would that be possible in 100 or probably 1,000 years or something like that?
Yeah, maybe I'm not sure I understood the question. Oh, okay.
Well, okay.
Yeah, okay.
Or probably the crossings actually was happened parallelly instead of serially. So one population is admixed of several gene pools, and then another population which is also already admixed in gene pool, and other somehow, and then these two population, there are something happens, and the progeny is combination of these two populations. It's already mixed, a lot of admix that can be the ancestry can be more than five or six population. This is also our discussion with probably actually I asked this to you yesterday, but probably my question is not that really clear. But now it's clear. And thank you, Pak Wicaksono.
Well, actually, there's a problem with that explanation. You know, if human bring those admix has to be parthenocarpic, because you know people would not bring those seedy banana everywhere they go. So if you're talking about the two admix that parthenocarpy and they cross in the wild, well, there's probably a chance too. But you know, it's I was thinking that would take a long time. That possibility is probably very small.
True. That's true.
I think there is still a lot to understand about the precise diversification process. Okay, so we have an idea or a hypothesis about a starting point, and even this starting point we need to understand a bit better. And then the whole diversification process is still something that we need to understand better. Yes. So this is we had now a general view of how the cultivars look like, and how exactly the diversification occurred is still to be studied and and characterized. Yeah.
Okay. Thank you.
Okay.
And Pak Wicak, is it okay?
It's okay.
Okay, thank you. So so there is a question in the chat box. So this will be the third question for you, Nabila. So this is question from Nurfianto Kiar Pangidung. How about the correlation about banana plant disease genetics with seasonal climate and irrigation regime?
Okay. I think I will leave this to Jean if he has an answer to that, because it's related to plant disease.
Okay.
I think he would be the best person to answer that.
Okay, so I will leave it. So okay, so for the third question will be from Bu Yuyu. Okay, please, Bu Yuyu.
Okay, thank you. I would like to know, are there any samples from Kalimantan or Sulawesi Islands in your research, such as Musa acuminata, subspecies of Microcarpa, and Musa acuminata variety of Tomentosa of Sulawesi? Thank you.
Yes. So in our setting, we had one representative of subspecies Microcarpa. I think it's the only one present in international collection. It's an accession that is called Borneo, I think. So that's the name of the accession.
Right.
And for Tomentosa as such, we did not have. That's something we did not have. And there is in the international collection one representative of subspecies Erans. It's called Agutai. It's, I think, from the Philippines.
Right. From the Philippines, right?
Yeah, exactly. And I think, Fajar, you told us that maybe Tomentosa was related to Erans, or I don't know if I'm correct about that. But we don't have the Tomentosa samples, no.
Okay, thank you.
Yes, just additional information for me that I include Erans, the collection from my supervisor Hugo. So the genetic is similar to what we call Tomentosa from Sulawesi. So yeah, we still have know exactly on much more accession just to make sure, because we know that the variation in the subspecies itself is actually can be more than we expected.